Hello everyone, I am Arnab Sadhukhan and today I am going to discuss about an enzyme, carbonic anhydrase. Okay, so as all of us know, um, our respiratory system takes oxygen from air, and this oxygen is transported through blood in the body cell, and the produced CO2 in the body cell is transferred through blood again to the respiratory system, and it releases CO2. So this CO2, so the carbonic anhydrase enzyme helps to hydrate this CO2, and it produces bicarbonate ion and H plus ion. Okay, and this reaction, um, it has a non-enzymatic rate constant 10 to the power minus 1 second inverse, and the same reaction in the presence of carbonic anhydrase enzyme, it has a rate constant 7 into 10 to the power 6, and it has a huge catalytic proficiency 7 into 10 to the power 7. And so carbonic anhydrase helps to make the acid base balance in our body, and this pH balancing activity of carbonic anhydrase supported by Bohr's effect curve, and increase of CO2 concentration leads to increase of acidity in the body, and consequently decrease of blood pH, and then the O2 binding affinity of hemoglobin decreases, as you can see in this curve. Okay, and there are some other functions also of the enzyme carbonic anhydrase. It produces acid in the stomach lining. In kidney, the bicarbonate ions control the water content of the cell, and in eyes also, bicarbonate ions influence the fluid balance in eyes. So the same enzyme also catalyzes the backward reaction in the lungs, where CO2 concentration is quite low, and this is the corresponding equation here. Okay, so this is the active site structure of carbonic anhydrase enzyme. Active site contains a zinc 2 plus ion at the center of this active site, and this is tetrahedrally surrounded by 3 histidine nitrogen, and the fourth coordination site is occupied by a water molecule, and the whole active site is wrapped by a protein chain. Okay. So to discuss the mechanism, first I would like to focus on the zinc 2 plus ion. For this zinc 2 plus ion, uh, here the wedge bond of this water molecule, sorry, of this water molecule is highly acidic, and the uh, water molecule surrounding the active site structure, they just snatched a hydrogen from this um, water molecule, and it lifts this OH structure, and then it can attack the CO2 molecule, and then it leads to um, this type of structure, and it produces a bicarbonate ion, and in the next step it produces a H3O plus ion, and then the total catalyst is regenerated. So after that, I would like to ask Dibyakanti Mal to present uh, his part.
Now, so far, 5 distinct families of carbonic anhydrase have been found: alpha, beta, gamma, delta, and tau, amongst which alpha, beta, and gamma, these 3 families have been extensively studied. Now, in alpha carbonic anhydrase, it is found in vertebrates and different algae. Human carbonic anhydrase also falls under this category. Now, alpha carbonic anhydrase is basically contain is the monomer where the zinc 2 plus is coordinated with 3 histidine moieties, and the fourth coordination site is definitely the occupied by water molecules. And there are 4 subgroups of alpha carbonic anhydrase: cytosolic carbonic anhydrase, mitochondrial carbonic anhydrase, secreted carbonic anhydrase, and membrane associated carbonic anhydrase. Now these four subgroups are basically the isoforms of the enzyme. That means they have the same function but they have different amino acid sequence. Now beta carbonic anhydrase, beta carbonic anhydrase is found in prokaryotic cell and in different plant chloroplast. Now beta carbonic anhydrase generally exists as different oligomeric state, within which two different types of structures have been found so far, type 1 and type 2. Type 1 beta carbonic anhydrase is basically dimeric, where the zinc 2 plus is coordinated with two cysteine moiety and one histidine moiety, and fourth coordination site is weakly bound with aspartate and water molecule displaces that. And in type 2 beta uh, carbonic anhydrase, this is a blocked conformation, and zinc 2 plus is coordinated with two cysteine moiety, one aspartate moiety and one histidine moiety. In gamma beta carbonic anhydrase Mycobacterium tuberculosis, the beta carbonic anhydrase shows the allosterism. That means the reactivity of beta carbonic anhydrase depends on pH. Dimeric at pH 7, this bacteria has the dimeric protein, where it is in the type 2 state, and as the pH increases it goes to the accessible active site, that is the tetrameric protein, and this is the type 1 beta carbonic anhydrase. Now gamma carbon, this is present in generally the methanogens. Now gamma carbon is basically trimeric molecule, where zinc 2 plus is generally located between the subunits, where two histidine unit and one histidine moiety of another subunit. And delta and tau, these are found in few diatoms and bacteria and chemolithotrophs. So at last I would like to acknowledge our course instructor, Professor Subhajit Bandyopadhyay, teaching assistants, and all my friends and classmates. Thank you.